are going to learn about capillary electrophoresis. In this chapter, I am not going to deal in detail of this topic. I am going to tell you in short capillary electrophoresis. These are the contents. Introduction, principle, mechanism, capillary electrophoresis steps and detectors, methods and modes, advantages and disadvantages, applications. Introduction. Electrophoresis is the motion of dispersed particles related to a fluid under the influence of a specially uniform electric field. Electrophoresis of positively charged particles is sometimes called cataphoresis, while electrophoresis of negatively charged particles is sometimes called as anaphoresis. Electrophoresis is a general term that describes the migration and separation of charged particles under the influence of an electric field. An electrophoretic system consists of two electrodes of opposite charge. Two electrodes are cathode and anode connected by a conducting medium called an electrolyte. Types of electrophoresis: zone electrophoresis and moving boundary electrophoresis. Zone electrophoresis consists of paper electrophoresis, gel electrophoresis, thin layer electrophoresis, cellulose acetate electrophoresis. Moving boundary electrophoresis: capillary electrophoresis, isotachophoresis, isoelectric focus, immunoelectrophoresis. What is capillary electrophoresis? Is a family of electrokinetic separation methods performed in sub-millimeter diameter capillaries and in micro and nano fluid channels. Which principle involved in capillary electrophoresis is: capillary tube is placed between two buffer reservoirs. An electric field is applied. Separation depends on electrophoretic mobility and electroosmosis. Using narrow bore tubes, capillary electrophoresis removes the Joule heating effect, which decreases band broadening, giving faster separations than gel. Capillary electrophoresis uses tubes 20 to 100 diameter and 20 to 100 centimeters in length. Is used with gel or without gel. Longitudinal diffusion is the main source of band broadening. Higher electric field results in high efficiency and narrow peaks. Types of molecules that can be separated by capillary electrophoresis. Which molecules is used in the capillary electrophoresis is proteins, peptides, amino acids, nucleic acids. Nucleic acids means RNA and DNA. Also unlike dual slab gel electrophoresis, inorganic ions, organic bases, organic acids, whole cells. Which mechanism involved in the capillary electrophoresis? How to works? Capillary electrophoresis works in two mechanisms: electroosmotic flow and electrophoretic mobility. Electroosmotic flow means movement and direction of the bulk solution. Electrophoretic mobility means separation of charged species. It is a principle for electroosmotic mobility. It consists of two buffer reservoirs, sample, anode and cathode, detector, capillary. Mm, Capillary is fused silica capillary, inner diameter 10 to 100 mm, length 40 to 100 centimeters. Capillary is polymer coating. Example, human hair. In capillary electrophoresis, which buffers are used? How much pH range? Vicine, phosphate, acetate, borate, etc. These are the formulas for basis of electrophoretic separation, migration velocity and electrophoretic mobility. Capillary electrophoresis steps. Electrophoresis is done in buffer filled narrow bore capillaries. Each capillary is about 25 to 100 mm in internal diameter, small cross sectional area, long length leading to high resistance, low currents. Depending on the charge, the molecules move through at different speeds. How separation is achieved? A suitable detector is then used to detect the solute as it comes out from an end of the capillary. The data obtained are analyzed by computer and represented graphically. Detectors used in capillary electrophoresis is types of types of detectors, representative detection limit. These are the which detector used to how much detection limits. Capillary electrophoresis consists of three types: moving boundary capillary electrophoresis studies the capillary electrophoresis, zone electrophoresis. Steady state electrophoresis consists of isotechophoresis and isoelectric focusing. Zone capillary electrophoresis consists of capillary gel electrophoresis, pre-solution capillary electrophoresis, capillary electrochromatography. Um, Pre-solution capillary electrophoresis consists of capillary zone electrophoresis, chiral capillary electrophoresis, affinity capillary electrophoresis, mesillar electrokinetic capillary chromatography. Common modes of capillary electrophoresis in analytical chemistry is capillary zone, capillary gel, capillary isoelectric focusing, capillary isotopophoresis, mesillar electrokinetic capillary chromatography. Advantages and disadvantages of capillary electrophoresis is it is simple technique, automated technique, high efficiency of separation, short analysis time, low sample volume, easy of operation. Ability to separate both charged and uncharged molecules, different mechanisms for selectivity, low cost to use aqueous rather organic solvents, hence environment friendly. Uh, what are the disadvantages for capillary electrophoresis? Um, the preparative scale separations, tricky compound species that are difficult to dissolve, reproducibility problems, aged, improperly stored blood samples, degradation products, abnormal hemoglobin, use other means of identification. Uh, Applications for capillary electrophoresis is, for example, capillary electrophoresis has been heavily studied within the pharmaceutical industry as an alternative to else in various situations. Detecting bacterial microbial contamination quickly using capillary electrophoresis. Current methods require several days. False positive are common simply by exposure to air. Techniques like ELISA, PCR, hybridization are specific to certain microorganisms. So, I have this is my effort to explain what is capillary electrophoresis. So, okay, I hope you understand. Thanks for watching. Drop a like, leave a comment, and don't forget to subscribe.